Am I audible? Hello? Anyone? Hello? Am I audible? Hello? Yeah, Naveen, am I audible? Hello, am I audible? Naveen, Rahul? Yeah. Sorry for being late. Actually, I had an experiment going on. So today's session might end a bit early. I'll try to cover some concepts based on the content, like what we taught yesterday, what I taught yesterday. Is my screen visible? Is my screen visible? Okay. So like this is regarding the week 7 thing, the usual class. So like I'll continue what I taught yesterday. So I hope you guys remember all the glutes and everything. So we were learning about gluco, sorry, not gluco, glycolysis. So we have right now we have covered about all the glutes, the transporters of glucose into the cell. So now we will continue with that. So consider this as a cell. So now from the blood, so from what we remember was that after after eating something, the glucose in the after the breakdown of carbohydrates from the GI tract, that is gastrointestinal tract, uh, monosaccharide, that is glucose, fructose, and galactose moves via the portal vein to the liver, where either it can be the storage or it is released into the blood circulation, and then from blood circulation. From the blood circulation, it goes to the cell. So think this as like now we have glucose molecules. So these are basically glucose molecules. So now tell me, for example, if this is SKM cells, what is the glut present here? What is the transporter present here? What is the type of transporter present here?
Group 4.
Glut 4. So glut 4 is present. Okay. Now, so when this glucose comes in, now the normal like glycolysis starts into the cell, and then that's it gets converted into pyruvate, right? So now we'll learn about each step like one by one. So now when this glucose goes in, and like if I say that like a lot of glucose comes in, like I mean since it's glut4, it takes a lot of glucose come like a lot of glucose comes inside the cell. Now like can this glucose escape back to the like I mean back to the membrane, like I mean back to the interstitial fluid? Can this glucose when the based on the action of so glut4 acts on like I mean insulin helps in intake of glucose in via the glut4. So insulin doesn't mind it, sorry. So insulin action is here, and then glut4 goes and binds to the membrane, and then a lot of glucose comes in. Now now when we take a lot of glucose inside into the cell, can this glucose go back to the interstitial fluid again? Because we have already learned that glucose moves from higher concentration to lower concentration. So if this channel takes up a lot of glucose in, that means the concentration of glucose is quite high here, right? So that means it will be low here. So that means it should go back. So can so the question is, can it go back? Not yeah, like I understand, not through good for like so like can it go back? So if so like if you're saying that okay, according to concentration gradient it does. So then how will this cycle proceed further? If it's first of all, GLUT2 is not present on SKM. I hope you guys remember that GLUT2 is present specifically in pancreas and liver. So I was asking, okay, so if it goes back, then how can this glycolysis proceed further? Obviously, it should escape back if the concentration is quite high into the cell. How to trap this glucose inside the cell? That's the question here. Anyone? By phosphorylation, yeah. So by phosphorylation, that's how it happens. Like it does. So once the glucose comes in. So basically, I'll write it here. Glucose is a six carbon molecule, right? Now when glucose comes in, it is trapped inside the cell by phosphorylation. So basically, the glucose is getting phosphorylated. It is getting phosphorylated, and that's out. And once it gets phosphorylated, why can't it go out? Anyone? Once there is an addition of glucose, why can't it go out? Um, Phosphorus is not like quite like it's not that heavy. It's like what's the molecular weight of phosphorus? I forgot, but I remember that it's not that heavy. So it's not the weight, obviously. Why can't this glucose escape back once it gets phosphorylated? I understand that okay, after phosphorylation it gets trapped in, but why is it trapped in? Anyone can guess? It's not about the concentration gradient. Okay, like, let me give you a hint. Uh, so what is the nature of what is the nature of phosphorus? Like, I mean, like, first, what's the nature of phosphorus? Nature in the sense, is it polar? Is it neutral? Is it negatively charged, positively charged? Like, what's the nature? What's the charge on phosphorus? Negatively charged, yeah. So this phosphorus is negatively charged. So this becomes highly polar, and polar molecules can't transport this thing back outside the membrane. Like polar molecules like does not transport like, I mean, does not get transport outside. One second. Where do I see the chat? Membrane transport won't allow phosphorus to go over. Yeah. So like since after the addition of phosphorus to this glucose, it becomes highly polar, and because of which this glucose can go back outside, like go back out the cell, can't go outside the cell. So that's the reason. Now let's go back to, so what's the enzyme, like what's like some enzyme should be present, right? Like now we are focusing on each step of the glycolysis. So what's the enzyme? Which enzyme phosphorylates glucose into glucose 6 phosphate? Hexokinase. Okay. Any other answer? Glucokinase. Hexokinase. And one is hexokinase and one is glucokinase. So like one thing, yeah, okay. First, I would like, tell me the difference between. So tell me the difference between um, two things: kinase and phosphatase. Phosphatase. So what is kinase and what is phosphatase? So tell me what is kinase first. It's a phosphorylating enzyme. Okay. So basically, kinase adds phosphate addition. And what about phosphatase? Removal of phosphatase. Okay. Removes. Can kinase act as a phosphatase? Can kinase act as a phosphatase? Like you told me that kinase can add phosphate. So I understand. Like, okay, that's the thumb rule. Okay, I understand. But can it remove phosphate as well? No. Anyone else? Anyone else? Okay, so this is like when I read about it, I too felt uh, quite a shock regarding it. So there is one exception to it, PFK2. Have you guys heard about this enzyme? PFK2. Any guesses? We are learning about glycolysis, so these terms might be, yeah. That's phosphofructose kinase 2. So this is a special enzyme. It basically has like the polypeptide chain has like two chains. I guess I don't remember exactly, 2 units or 2 chains. So one of the chain acts as a phosphatase, while the other acts as a kinase. So basically, if for example, if this is the enzyme PF PFK2, one acts as a phosphatase. It will remove the phosphate, and other one on the other hand, basically think as other hand, it will act as a kinase. So this is one of the exception of kinase enzyme. Even though it's a phosphofructokinase 2, the enzyme name, it can act as a both phosphatase and both kinase. And this enzyme, when to act which one, it will be dependent on the substrate that is present. So like will be like it's a part of the glycolysis. I think most of the textbook don't talk about this, but like I will. So we'll be discussing in this the same I mean the same pathway. So you guys should remember that. Okay, there are two things, kinase and phosphatase. Kinase is basically like as a thumb rule, like kinase adds phosphate to the substrate, while phosphatase removes phosphate. And there is one exception, that is phosphofructokinase 2. It can act both as a phosphatase and kinase. So I'll tell you like when it comes. Okay, now this is done. Now we were focusing about hexokinase and glucokinase. So basically it adds phosphate here, so addition of phosphate, right? So now, is hexokinase and glucokinase same? First question is that, like, are they synonyms, are they same, or like are they different? Anyone? Hexokinase general term. Hexokinase general term, whereas glucokinase is specific. Okay. So are you telling me both their function are similar? And if you're telling me a glucokinase is specific, so I'm asking you what it is specific to. Like, what's its specificity? Like, in what terms you're telling that it's specific? In what term it is? Okay. So hexokinase, like, I mean, you're telling me that hexokinase is not specific. Okay, let's start with this thing. So yesterday we learned that when we digest complex carbohydrates, there are three monomers that arise. That is glucose, fructose, and galactose. Right. Now tell me. Now tell me. Uh, um, these two enzymes act on which substrate? Do they are like are they specific to only glucose? Are they specific to some fructose or galactose, or are like equal to all? Like they can take everything. First, tell me about hexokinase. Is it specific to something, or like it can act on all of them? So specific to hexose sugar. Okay. So glucose is 6 carbon. Okay. Hexose. What about fructose? Is it like what's the carbon number of fructose and galactose? Okay, like fructose and galactose, how many carbons are there? Six, six. Okay, and hexose means six. So you're telling me that hexokinase is specific to everything, right? I mean, like it can use all of them, right? Based on the what name suggests. Yes or no? Okay. So basically, hexokinase works on all the substrates. It can utilize all of them. So it acts on glucose, fructose, galactose. As the name suggests, hexo, it can take all the six carbon sugars and do the and phosphorylation. It can act on that. Now tell me about glucokinase. Can it act on all of them? If you're going by name, it's like if it's glucose. Like, do you think it's just specific to glucose? So based on what name suggests, it is specific to glucose, but no, it can take up all of this like fructose and galactose, all of this. So basically, irrespective of what's the monomer, like what's the monosaccharide coming in, if they can phosphorate all the three forms of monosaccharide, that is glucose, fructose, and galactose. Even if the name is like I mean quite deviating, misleading, but it's not. It can do like it can phosphorate all of them. Like hexokinase and glucokinase can phosphorate all of them. So now this thing is okay. We have done about the substrate. It can act on all the three monosaccharide. Okay. Now let's go back to what is hexokinase and glucokinase. Okay. So like these are like one of most in like first of all, whenever in a pathway when you see a single forward line or like single reverse line anything, it's a rate limiting line because this enzyme determines the rate of that pathway. So if you see here the single line, this is the first. Mostly in a lot of exams, they ask you like how many rate limiting steps are there in glycolysis. So here, this is the first rate limiting step. Okay, so these enzymes are quite like I quite not quite like it's very important. So let's discuss about these two, hexokinase and glucokinase. Okay, so first of all, we have learned that they act on all the substrate, all monosaccharides, right? All monosaccharides. I mean the monosaccharides. Okay. So the first point. Next thing, hexokinase and glucokinase. First, let's talk about okay, let's start with this thing distribution. Um, first, I'll tell you that okay, both of them act on monosaccharides. Okay, they both phosphorylated, both are kinase enzyme. Okay. Now next, we talk about the distribution. So like, do you think like all the cells have hexokinase and glucokinase both? First thing, they are different. Like they are different. Like even they're like hexokinase is also known as sorry, glucokinase is also known as hexokinase B or D. I don't remember the exact name. It's like something like that, B or D. I think it's mostly D. And this hexokinase is also known as glucokinase 4, I think. So I have forgot. Okay, it's called D. Okay, thank you. And this hexokinase is called I think glucokinase 4. I exactly don't remember, but something like that. So first thing is they are both different in terms of efficiency, distribution, and everything. So let's discuss about that. So first thing is like where do you think they are like found? Okay, let me like before distribution, I'll tell you about the efficiency first. Okay, that will help you to determine where it is located. So hexokinase. So hexokinase is basically if I tell you the graph thing, if enough cell like when we are like eating like a lot of it, so slowly like the concentration of glucose is increasing, right? So when this is the graph of hexokinase. So as we are like we keep eating. So basically, the graph. Like I hope you guys have learned about Michaelis Menten thing. So so this is the hexokinase, the enzyme activity of hexokinase. So if the number of glucose is increasing, the activity of enzyme is increasing, and then it goes like this. So basically, till a point when the glucose is available in the body, like from the lowest to the highest. So when it's low and then it's gradually increasing, the efficiency of this enzyme to phosphorylate this glucose to glucose phosphate, glucose 6 phosphate, it keeps on increasing. But after a certain point, even if you increase the glucose concentration, there won't be any activity. Basically, this enzyme has been saturated. Okay, so this is the hexokinase. And the same thing, when we give glucose to hexokinase. Initially, when there is a gradual increase, there is a like slow increase with this because it does not get activated with the low concentration of glucose. But as we increase the glycolysis, I mean like glucose concentration, the efficiency of the enzyme keeps increasing. So this is glucokinase. So this is the first difference here. So first, let's discuss the concept of Km here first of all. So can you tell me what is Km?
Substrate concentration at maximum velocity is attained.
Okay, yeah. So it's the substrate concentration at which half of the maximum velocity is reached. Okay, so when we talk about hexokinase, the Km of this hexokinase is quite low. So if this is Vmax, so Vmax is like half of the Vmax is here, and this is Km. And again, for this glucose, the Vmax is like basically unattainable. In most cases, it's unattainable. So the Km for this thing will be quite high. So based on the Km, can you tell me which enzyme? Like first, let's write here. The Km is low for this, and Km is high for this. Vmax is quite high for this, and Vmax, I think it's constant or low, mostly low. So tell me like which based on the Km, can you tell me like which has the higher affinity of glucose? Yeah, sorry, sorry. Yeah.
Hexokinase.
Hexokinase, it's not low. It's like maximum is like Km is low, but the maximum it reaches fast. Yeah, sorry. So hexokinase affinity is more. Affinity is high, and this is affinity is low. Based on the Km, we can decide that, right? So now let's talk about the first. Let's revise this. Hexokinase and both glucokinase work on all the monomers of carbohydrate. To that they convert from one form to the phosphorylated form. Second thing, the efficiency of hexokinase and glucokinase. As we increase the concentration of glucose, initially the hexokinase can easily convert glucose into glucose 6 phosphate, but later as we increase the concentration, it gets saturated fast. But glucokinase does not. So therefore, its Km is quite high, and then the Vmax is also high, very high, like in terms of with respect to hexokinase. Now I'll tell you a scenario. Okay, let's talk about the distribution. So now when we are eating, when like we are having some carbohydrates, slowly glucose is released uh, uh, from the circulation to into the cell. So which do you think? Which thing in normal cells? Which enzyme will be present most prominently, whether it be glucokinase or hexokinase?
Glucokinase.
Glucokinase. Okay. Why? Why not hexokinase? Sorry, you told glucokinase. Okay. Um, Glucokinase. First thing, let's go by this. I'll open up a new page. We're talking about hexokinase and glucokinase. So now we were discussing about the distribution. Okay. First, this is the main point here. Km is low, Km is high, very high. So you saw the graph of hexokinase and glucokinase. The graph of hexokinase was this X, and glucokinase was like this. So when you're eating a lot of carbohydrate, then I understand that okay, there will be a lot of glucose in your body, and then it will easily be glucokinase can be easily activated. As you can see, there is a small delay here. So that means it needs enough glucose. Then it needs enough glucose to get activated and then start its function. But here, um, there is like a sudden rise of the activity here. So that means like what if you don't eat for a while, like you're in a fasting state? That means the glucose is quite low in your body, and do you think then you will get energy if glucokinase is present on all your cells? When there is low glucose in your body, then how can you get energy? It is quite like it's not that possible. Like it's basically not possible in that way. So that's why this glucokinase, since it needs a lot of substrate to get activated and then perform its function, it is not present in all the cells. It's the hexokinase. It is ubiquitous towards all the cells. So that even if there is a small amount of glucose coming into the body, it can easily take in, it can easily trap inside the cell, and it can go further into the glycolysis pathway. So hexokinase is present all over the body. Okay, now as I told you that glucokinase, it needs lot of glucose. So where do you think it is present?
liver cell.
Liver, yeah, because liver is in direct contact to our GIT. GIT monosaccharides comes in from there. It goes to liver first. So in liver, it in order to perform this, like glycolysis to occur, uh, glycolysis to occur, it has to trap a lot of it has a lot of source of glucose here. So in order to perform this, it needs glucokinase, and not hexokinase. So this glucokinase is specifically present in hepatocytes. So that's one difference. Hexokinase is present on all our cells because liver does not give a lot of glucose outside to the blood circulation. It gives based on its necessity. So not all cells require glucokinase. It requires hexokinase because they can work at the basal level. But in liver, there is initially itself there is a lot of a flooding of carbohydrates. So this lag is not that prominent there. It just shows a singular curve, like singular line. So remember that glucokinase or hexokinase D is present in hepatocytes, liver specifically. Okay, so now we have learned about the distribution. Next, next what we can say is okay. Three. Okay, I think we have covered all the points in hexokinase and glucokinase. Okay, this thing's covered. Now, as I told you that this is the rate limiting step of the first step is the rate limiting step. So we were talking about glucose to glucose 6 phosphate, right? And here or hexokinase works in normal cells, and glucokinase works in hepatocytes. Okay. Now, since this is a rate limiting step, that means this process should not be always if there is already like lot of generation of ATP. So why does the cell need to utilize this? It can store this glucose can be stored in glycogen. So in order to stop this process of like further going into the glycolytic pathway, these enzymes need to be stopped. So what do you think will stop this enzyme? How will the cell know that okay, it is there is a lot of glucose already inside it, and I should not continue with the glycolytic pathway? How will that cell know? How will the cell inform this enzyme it is enough? You just stop phosphorylating it. This conversion of glucose to glucose 6 phosphate is a rate limiting step. It is a very regulated and crucial step in a glycolytic pathway. So mostly in order to stop this enzyme, this product will, if there is this, the product of this is like in quite abundance, it will act as a inhibitor, and it will stop this enzyme, like in this enzyme. So, so will it stop both the enzyme? Will abundance of glucose 6 phosphate into the cell stop both the enzymes, that is hexokinase and glucokinase? This is a product based inhibition. The product of that enzyme is inhibiting the enzyme in order to stop the one second, I have charge issue. Anyone? Will the glucose 6 phosphate both stop both the enzymes, both hexokinase and glucokinase? So first thing, glucokinase is like a, is my screen visible? Hello, is my screen visible? Yes, okay. So this product inhibition, it's only for hexokinase. That glucose 6 phosphate inhibits hexokinase. So what about glucokinase? As we all know that glucose kinase is specifically present in hepatocytes, its function is much more important because there is a higher flood of because there is a higher flood, because there is a higher flooding of glucose. Can we do that? Sorry, it is highly like compared to hexokinase, it is highly regulated. So what is the next step of after glucose 6 phosphate? What is the next product? Fructose 6 phosphate. So we have learned about glucose to glucose 6 phosphate and then to fructose 6 phosphate. So this is a single step. It is governed by hexokinase or glucokinase. So we have already talked about glucokinase and hexokinase and glucokinase. And the one thing now we are mentioning is the inhibitory. Like what does like who stops hexokinase and glucokinase? Hexokinase is stopped by glucose 6 phosphate, while glucokinase it is present in liver and it is highly like much highly regulated in terms of the inhibition as well. And this glucokinase is inhibited by fructose 6 phosphate. It is not inhibited by glucose 6 phosphate. It is inhibited by fructose fructose 6 phosphate. Okay, so you guys have to remember this point really like I mean like properly. First of all, that hexokinase and glucokinase are not same. They differ in their activity, they differ in inhibition, they differ in the distribution as well. So this is inhibited by fructose 6 phosphate, and this glucose kinase, like as I told you, it is present in hepatocytes, right? So where do you think it is present? This hepatocyte. Where do you think this thing is like? We all know that glycolysis occurs in the cytoplasm. So, like you mentioned yesterday, that some of you, like yeah, all of them, all of you, I guess, that all the enzymes are present in cytosol, right? So, do you think that glucokinase is also present in cytosol? Yes or no? Anyone? Okay. When first, so I told you that glucokinase is always activated when there is an abundance of glucose in it. So normally, when there is no when we are in fasting state, or like we have not eaten for a while, glucokinase is not activated, and when it is not activated, it is inside the nucleus. So remember this. This glucokinase is first in nucleus, and when there is like minimal amount of glucose, hexokinase will come, like hexokinase will be active and it will take some glucose in, and then the process of glycolysis occurs. But glucokinase is only active when there is a lot of glucose into the body, like when it lot of glucose comes into the hepatocyte, like liver cells. So but before that, in inactive state, this glucokinase is present in the um, nucleus, and this is held by a protein called glucokinase regulatory protein. Glucokinase regulatory protein. So this is it. So when does it become active? When there is abundance of glucose, and then glucose comes in. Glucose even goes into the uh, nucleus. It brings this glucokinase. It basically senses that yeah, glucose is so much that it is coming inside into the coming into the nucleus. So this will inform this, like once glucose goes in, it this glucose will bring glucokinase outside to the cytoplasm. Glucokinase comes outside, and then the process of this thing occurs. Okay, and this enzyme is inhibited by fructose 6 phosphate. So this is one of the crucial main point. Normally in inactive state, glucokinase is in nucleus, then held by glucose kinase regulatory protein. Then glucose comes in, glucose activates it and brings it to cytoplasm. Cytosol. Is there any doubt here? If you have, you guys can ask. Okay. So always remember, glucokinase and hexokinase are not same. So I hope you guys have like noted down all the differences here. Okay. Now only this much was there. Okay. For this, there is like these are the main important differences between glucokinase and hexokinase. Now this glucose 6 phosphate is converted into fructose 6 phosphate. And what is the enzyme here? What's the enzyme involved in this? Yeah, phosphofructose like isomerase. Yeah, like I don't remember. Okay. Yeah, it's not isomerase right now, I think. Okay, it's shifting. Okay, it's isomerase. It's phosphofructoisomerase. Isomerase is like enzyme that can shift. So as you know that these are also, this is also 6 carbon and this is also 6 carbon. So basically the chain, like there is some shifting of hydroxyl group in order to form glucose to fructose. So phosphofructoisomerase acts on it. So this is not that important step. No one will basically ask about this. Okay, now next step. When someone asks you which is the most important step in glycolysis, you have to answer this. Fructose 6 phosphate 2, fructose. What is the next product? Fructose. What is it? Fructose
1,6 bisphosphate.
1,6 bisphosphate. Now tell me one thing. What is the difference? Like, are this both same? 1,6. Okay. If I write 1,6 biphosphate, is it right? Which one is right? Is it like 1,6 bisphosphate or 1,6 biphosphate? Like, okay, it's not right. It's not right. Yeah, it's bisphosphate. So tell me, what is the difference between these two? What is bisphosphate and what is biphosphate?
When two phosphate attached to the same carbon, then it's biphosphate. And if they are attached to the different carbon, then it's a bisphosphate.
Okay. So okay, let me tell you this. I forgot. But still, what I read was. So tell me, this is bis or bi? The first one is bis
Bis,
for not present continuous. Okay, first one is bis or bi?
bisphosphate.
Um, what was it? Papain. Papain is right. We use bis only when they are not continuous. So when the phosphate group or any group is in like continuous, like they are on the different two different carbons, but they are not they are continuous. So when there is like some kind of group is attached and that two continuously, it is called bi. It is not bis. And when like it is on some like separate carbon but far apart, they are not continuous, then only bis comes in. So remember this difference. Bi is separate carbon but continuously, while bis is separate carbon but it's not continuous. It is like one gap is there, two three, or like I mean like it is basically separated. So that's you should remember that the group present continuously, and this thing group is not continuous or far apart. You should always remember this thing. Okay. So we were talking about. So I was telling you, when someone asks you what is the most crucial step of glycolysis, you should always answer. It is the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. This step is so highly regulated that it can determine whether the glucose like I mean glycolysis should proceed further, like it should slow down, it should faster, like it should like be fast, or it should go in reverse. Everything is determined by this step. This is the most regulated and most important crucial step of glycolysis. So always remember, it's the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Okay. Now when we are talking about this is fructose 6 phosphate to fructose 1,6 bisphosphate. Now please tell me the enzyme that is involved here. What is the enzyme that is involved here?
Phosphofructokinase.
Phosphofructokinase. So do you guys remember that we have talked about phosphofructose before, like just a while before, like I mean around 20 minutes before. So which phosphofructose was that?
Two PFK2.
PFK2. So do you think this is PFK2? Can anyone tell me the what's the exact answer? Like which thing is this? This is PFK1. PFK, it was PFK2. Never get confused. PFK2 that can act as kinase and phosphatase. So this is PFK1. Okay. Okay. So when this PFK is active, it converts. It basically adds one bisphosphate, like one more phosphate group um, to the fructose chain. So that's a simple answer. But how is this regulated? So this again, this is the second rate limiting step. Remember this as well. This is the second rate limiting step. This determines whether how fast is the glycolysis going. It should go further. Like I mean, like the fast, like I mean the rate basically. Rate, rate, second rate limiting step. So the first, what was the first rate limiting step?
Glucose to glucose 6 phosphate.
Yeah, glucose to glucose 6 phosphate. Okay. So this is the second rate limiting step. So now we read that. Okay, tell me this. What is the like which product stops glucose kinase? Like who stops glucokinase to convert glucose into glucose 6 phosphate?
Fructose 6 phosphate.
And what about hexokinase?
Glucose 6 phosphate.
So always remember that difference. Now, this fructose phosphofructokinase 1 is extremely regulated. So you can see these points. So these are the regulatory points that I have drawn. So when the cell is proceeding further, like into the glycolytic cycle. So what is the first main product that we get from glycolysis? We get energy, right? Energy in the form of.
ATP and NADH.
So like okay, we are not discussing about NADH because there is no point here. But like first thing is ATP. Okay. Now when this ATP, like when we utilize this energy, what is the next form? Like ATP is broken down into like okay ADP and then to AMP. AMP is the final form. Okay, next these two. Now there is this one more product of Krebs cycle. It's citrate. Okay, let's discuss about these three first. So AMP, ATP, and citrate. Now, when just tell me this, okay? Fructose 6 phosphate is getting converted into fructose 1,6 bisphosphate. So if there is a lot of ATP, like once this glycolysis is occurring continuously and there is a lot of ATP in the body, like in the cell, should this glycolysis proceed further or should it stop? When you already have so much of energy, do you think glycolysis should occur? Anyone? No, it should not occur. Yeah. So that's the thing. ATP, one of the site here is ATP. When ATP is locked in the cell, ATP comes and binds to this pocket and tells PFK that okay, there is like lot of energy in the body. You have to stop converting phosphate 6 phosphate, fructose 6 phosphate into fructose 1,6 bisphosphate. So this is a negative regulator. Now, where do we find citrate? Citrate comes up in which cycle?
PCA.
Okay. So when this thing comes up, when citrate, basically, when there was lot of glucose was converted into pyruvate, and this pyruvate went to TCA. There comes the citrate. So when there is lot of citrate, it again goes and binds to this PFK1 and tells that there is like lot of already like I mean there is already lot of conversion of glucose to pyruvate and pyruvate to citrate, so you have to stop. So this citrate is again a negative regulator. When these two comes and bind to this PFK molecule, when they are in abundance in number, like into the inside the cell, they come and bind to these pockets, and when they come and bind to these pockets, they basically inhibit this. So when they come and bind to this PFK1, there will be no conversion of fructose 6 phosphate to fructose 1 6 bisphosphate. Okay. Now, third, we have talked about ATP and citrate, which are negative regulator. Always remember they are the negative regulators. Now, when ATP is broken down into AMP, that means the cell has utilized almost all the energy that we had, right? So this when there is like lot of AMP into the body, this AMP comes and binds to uh, this phosphofructokinase 1. It comes and bind to this, and it tells the cell that okay, there is like no energy left, everything is utilized, now please proceed with the glycolytic cycle. And this is a positive regulator. So we have learned about ATP and citrate and AMP. So these are basically, these two are negative regulators and these are positive regulators, right? Understood? Any doubt until here? Hello? Is it clear? Like, do you guys any have any doubt? Like, I mean, so it was like it's just a simple concept that when this glycolysis occurring is going ahead, there is like accumulation of ATP and citrate, and this when they are like in larger number, they come and bind to this PFK1, and they basically stop it. And when all this energy are utilized, all this ATP are utilized, all this citrate are done. So when there is a higher concentration of AMP, it comes and goes. It goes and tells the PFK1 that we need energy. Please make some energy for us. So that's a positive regulator. Okay. Okay. Now we have talked about PFK1. What happened in RBCs? So RBC follows like it does not require that much energy. So basically, the conversion of glycolysis, glucose to pyruvate itself, when there is a like a release of I think two molecules of ATP, that is enough for that. So the lifespan of RBCs are basically 3 days, I guess, uh, as long as I remember. But yeah, the like the energy is good enough for that. So they don't need the TCA cycle and everything. Okay, now we have one more pocket left. Hmm, Forgetting something, I guess. Fructose 6. There is one more positive regulator of this this pathway, I mean this reaction, and it is fourth fourth one is fructose 2,6 bis phosphate. Have you guys learned about this? Fructose 2,6 bisphosphate. Have you guys heard about it? So we are here like convert using fructose 1,6 bisphosphate that goes down into the cycle. Have you guys heard about fructose 2,6 bisphosphate? No? Okay. So we have learned about phosphofructokinase 1 that basically converts fructose 6 phosphate into fructose 1 6 phosphate, and I have already told about FK sorry, PFK2. So that's where it comes into play. This when there is like abundance of fructose 6 phosphate into the cell, some of this fructose 6 phosphate is converted into fructose 2,6 bisphosphate by PFK2. So when this enzyme converts fructose 6 phosphate to fructose 1,6 bisphosphate, there is the conversion of fructose 2,6 bisphosphate. Bisphosphate. So here the enzyme is PF PFK PFK2. Say ATP binds, AMP binds, positive. Here citrate binds, it's negative. And again, which thing was left? What was the third thing? Hello? PFK, sorry, yeah, sorry. It's PFK1. So, like, what is the third thing here? It's ATP. Now we are talking about the fourth positive regulator, like second positive regulator. So, this is converted by PFK2. So, this PFK2, this PFK2, so this PFK2, when there is like a lot of fructose 6 phosphate in the cell, this some of this fructose 6 phosphate is converted into fructose 2,6 phosphate, and it again tells the cell that there is like a lot of glucose coming in. You have to convert it to energy. So this is the fourth positive regulator. So right now we have learned there are like two negative regulators. That is ATP, when there is like, this is a logical thing, that ATP and CMP, that is when the glycolysis has proceeded further, this glucose is converted into ATP and citrate, and they will go back and stop this PFK1, telling that there is a lot of energy. Please stop converting fructose 6 phosphate to fructose 1,6 bisphosphate. So this is the first negative regulator, and the positive regulators are AMP. When all the energy in the cell is utilized, all of them are converted into AMP. It will go and tell the enzyme that okay, convert more. And second thing is phosphate 2,6 bisphosphate. Bisphosphate. Now there is a lot of things special about PFK2. So I was telling that PFK2 can act as both kinase and phosphatase. Now, who determines that whether this will act as a phosphatase or kinase? Who do you think determines it acts as a phosphate or kinase? Anyone a random guess or anything? They are learning about glycolysis. Glucose concentration, and what determines the glucose concentration? What what determines the glucose concentration into the cell? Go back to the hormone level. What are the two hormones that regulate glucose concentration into the cell?
Insulin.
and glucagon, right? So yesterday I had mentioned about GLUT4, how it how insulin acts on this. So this is basically a RTK receptor, and this is a insulin receptor, and this is a glucagon receptor. Okay, okay. We are learning about PFK2. This is one that things it is think as one site of activation. So half of it is kinase, and half of it has phosphatase domain. Now, okay, first thing. Okay, let's talk about when there is very low glucose in the body. Which do you think will get activated? Which do you think will be released? Whether it will be insulin or glucagon? When there is low glucose into the body, glucagon. Okay. So when there is glucose low, glucagon is getting active, and when the glucose is quite high, insulin will get active. Okay. So now the condition is low glucose. So when there is low glucose, the glucagon will come and bind, and this thing will give signal that there is already less glucose into the body. Please don't utilize this glucose further into the glycolytic cycle. So when there is low glucose, glucagon will come. Glucagon will come. It will bind to the glucagon receptor. It will give signals, and when if someone has learned about this is a G stimulator. It is a GPCR, G protein coupled receptor. So it's a G stimulated protein. It will go and activate adrenal. Like right now, I don't think you guys have learned about it. I guess I don't know. So it will activate adenine cyclase, and adenine cyclase will convert ATP to CAMP. CAMP will activate phosphokinase A. So this is one of the major enzyme. This controls a lot of pathways. It activates, deactivates a lot of enzymes in this thing. So when this PFK is getting activated after the addition of CAMP goes and binds with the sites, and PFK is activated. PKA is activated. That is uh, protein kinase A. It's not phosphate. It's protein kinase A, and this is adenyl cyclase. Adenyl SY cyclase, and this converts ATP to CMP, and CMP goes and binds to this protein kinase, and it gets activated after phosphorylation. After addition, after binding of protein CAMP. So this PFK, what it does, it is a kinase. So what its major role will be? Addition of phosphate. So it goes and binds this phosphate here. Okay. When this phosphate is added by PFKA, okay, should not use. Okay. So when this phosphate what is the PFK is enzyme is acting on fructose 6 phosphate to fructose 2,6 bisphosphate. This will basically speed up the process. Okay, one more thing, that this is a very strong regulator of glycolysis. Like when it goes and binds to the PFK1, this is that enzyme PFK1. When it goes binds to this F, when this F26 phosphate goes and binds to this thing, it is a extremely really strong positive regulator. This thing will increase the glycolysis in lot of folds. Like this will increase a lot compared to any other, compared to AMP. This will regulate it really fast. So the rate will be really increased when this F26 goes and binds. So when this PKA goes and phosphorylates this enzyme, that is PFK2, it will activate, sorry, it will stop the kinase activity and induces the phosphatase, starts the phosphatase activity. Phosphatase start. So basically when this phosphatase gets activated, this fructose 6 phosphate is back converted into fructose 6 phosphate. Okay, this is the condition when there is minimal, basically when the body is running on low glucose concentration. When there is low glucose concentration, glucagon will get activated. Basically, we need much like more glucose in the blood. The glucose in the blood should be maintained almost like not almost like every time. So when there is low glucose in the blood, this glucagon will get active. It will signal the cells to not use this glucose and just to leave it out. Like I mean, like to get it out. So during that time, this kinase goes and activates this PFK phosphatase domain. Phosphatase domain. So when this phosphatase domains get activated, it removes the phosphate from here, and it again converts it to fructose 6 phosphate, and therefore, as I told you, this fructose 2 6 phosphate is the strongest regulator of this PFK1. In order to increase the speed of the glycolysis, this thing is gets really slow and basically nil. Not nil, basically really slow. It won't consume that much of glucose. So this thing. Now, second condition is when glucose is very high. Now when it's very high, the insulin comes in. One second. Sorry, like, am I audible now? Sorry. So we were so one of the like we have already discussed when the glucose, it's like everything here is based on logic. So when glucose is quite less, then the cell should not use glucose, it should give it back to the blood, because the maintenance of glucose into the blood is quite like really, extremely important. Therefore, so when glucose is low in blood, the glucagon is secreted, and this secretion of glucagon, it goes and bind to the glucagon receptor, that is a GPCR. Maybe in the higher classes you might read it, and specific GPCR is GS. So GS is basically, S stands for stimulatory. So when the GS get activated, GS goes and activates adenyl cyclase. It is found on the membrane. So this adenyl cyclase converts ATP into CAMP, and this CAMP goes and binds to PFK, sorry, PKA, and then PKA gets activated, and it activates the phosphatase enzyme. Sorry, phosphatase domain of PFK2. So this was that phosphatase domain of PFK2 is activated, and then for the glycolysis will not proceed further. Basically, the glycolysis will get slowed down, or like it won't proceed further, and the glucose will be intact. So this was one of the pathway. Now second thing is, what if the glucose is high in the cell, like high. So when the glucose is high in the blood, the insulin is secreted, it goes and binds to this IR, like insulin receptor, and this is a like type of RTK. This is hmm, RTK was tyrosine kinase, receptor tyrosine kinase, yeah. This was GPCR. I don't know like if you guys have read it or not. So these are the type of receptors that are found. This is RTK, that is receptor tyrosine, that is amino acid tyrosine kinase, and this is G protein coupled receptor. So these are the two different receptor. One of the two different receptors. So when this insulin gets activated, it activates lot of things, and one of this regulator is, it activates, it also activates one kinase. No, it activates a phosphatase. It activates a phosphatase. So glucagon activates a kinase. This insulin activates a phosphatase, and it will remove this. It will remove this phosphate. And when this is removed, again the kinase activity is activated. So always remember, when it's there is no rule that when there is activation of when there is addition of phosphorus, the enzyme will get activated. Sometimes, while when we add phosphate, enzyme can also get deactivated. In some cases, when there is a removal of phosphate, it can get activated or deactivated. Initially, when I used to read, like when I was in lower classes, I used to think that okay, when we add something, like when we add phosphorus to any enzyme, that enzyme gets activated and then it does the function. But later, I came to know that it's not always the case. As I told you, that PFK2, like it can act as a phosphatase as well. So there is not much information in the lower classes, but yeah, when there is addition of phosphate to any enzyme, it does not mean that enzyme will get activated all the time. It can sometimes leads to deactivation. One of the most common example is lipase enzyme. I don't remember the exact. I think it was I forgot, there is certain lipase that once it gets, okay, I forgot the name. There is this lipase, when it gets phosphorylated. Um, when it gets phosphorylated, it becomes inactive. So there is no rule that phosphorylation will lead to activation. Okay, now we have covered till, I think I have given all the information here. So like, let's one time. Actually, I have an experiment planned right now, so that's why I can't continue much. But I had to discuss a lot of questions. Okay, like I'll just complete this thing. So what we have learned till now. So I just want to give a summary so that you guys don't forget. Um, this was glucose. So what glucose is converted into glucose 6 phosphate. So this phosphate addition is to trap the glucose. So always remember trapping of glucose, because phosphate is highly polar and it can't it won't be able to diffuse out again. Trapping of glucose. This step is a rate limit. This is the first rate limiting step. Limiting, and this is done by, like, if you guys are there, please type things in the chat box so that I know that you guys remember these things. So this is hexokinase and glucokinase. There are a few points that you need to remember. This is hexokinase ubiquitous, um, low Km, higher affinity. Um, what else? Was there anything special about this? Did I miss something? So this is hepatocytes, high Km, low affinity, present in nucleus. Glucose activates it. Yeah, fructose 6 phosphate. No, sorry. Yeah, sorry. Which was the, like, what was the inhibitor here? Which stops hexokinase?
Glucose 6 phosphate.
So the inhibitor is glucose 6 phosphate. Here it is fructose 6 phosphate. Now, did I leave anything important now? So hexokinase is also known as glucokinase 4. Please cross check this fact because I did learn about it like a really long time ago. And this is also known as hexokinase D. Okay. So never get confused. They are not the same. Their role is similar. They phosphorylate glucose. They act on all the monosaccharides. All the monosaccharides. This glucokinase is very specific to hepatocytes. Its Km is quite high. Therefore, when there is a lot of, like, abundance of glucose into the cell, then only it gets activated. Because when there is a lot of glucose, glucose goes into the cell, then again to the nucleus, then it brings back the glucokinase. The glucose kinase is regulated by glucokinase regulatory protein present in the nucleus. And when glucose goes in, it removes this and it takes the glucokinase out. And then the things happen, the phosphorylation happens. What else? What else? What else? Okay, this was the main difference. Okay, now glucose 6 phosphate was converted into fructose 6 phosphate. It is a both double this thing. This isomerase works here. Now the most important step. Always remember fructose 6 phosphate to fructose 1,6 bisphosphate is the most important regulated step of glycolysis. This is fructose 1,6 bisphosphate. The difference between bisphosphate and biphosphate is that bis means it is far apart, while bi is they're in the continuous thing. And the enzyme that works here is uh, what is the enzyme here? Phosphofructokinase.
One.
Which thing was that? Just one. And here there is a small enzyme called fructokinase 2 that has a domain of both kinase and phosphatase. And what are the positive regulators of phosphofructokinase 1? Tell me the positive ones.
AMP and fructose 2,6 bisphosphate.
AMP and fructose 2,6 bisphosphate.
Citrate and
What are the
ATP.
negative ones? ATP and citrate. Okay. So am I missing something here? No. Okay. And what is the strongest regulator of PFK1? So it's F26P. This is the strongest regulator of this enzyme, and this step is important, and this step determines the rate of the glycolysis. Okay, now next is next. What was that? Next one was about this phosphofructokinase 2. It can act both as kinase and phosphatase. Um, so it basically converts when there is like a lot of fructose 6 phosphate into the inside the cell, it gets converted into fructose 2,6 bisphosphate, and this 2,6 bisphosphate it goes and regulates the this PFK1, and this enzyme the cell determines when it can act as a phosphatase and when it can act as kinase. It is determined by the glucose concentration into the blood. So when there's glucose concentration, when there is high glucose concentration, insulin gets activated. Insulin will activate one phosphatase, like it will go after binding with insulin receptor phosphatase, and this phosphatase will remove the phosphate from PFK2, and uh, the kinase activity will proceed further. Kinase activity will be high. While when there it's low, when it is low, when it is low, glucagon is released. Glucagon goes and binds to glucagon receptor. Glucagon receptor activates PKA, that is protein kinase A. It is a kinase. It adds phosphate, and addition of phosphate to both the domains will activate. It will deactivate kinase, and activate phosphatase. So this is basically tells the cells that there is not much glucose left in the blood. Please stop this reaction coming towards the forward thing. Is there like, do you guys have any other doubt? Anything you want to ask related to this thing? No? Okay. So I hope you guys understood. Like till this, I think I'm teaching a bit slow. So I'll try to. Actually, I have an experiment planned now, so I think I need to leave. But next time, like I had, do you guys like want to continue with this? I mean, like, do you want me to teach like this, or like, do you guys prefer just the question answer session? Actually, like whatever I'm teaching here, like as a PMRF thing, I have to upload it in YouTube. So if you guys need to revisit anything, you guys can check up these videos. Like whatever I'm teaching here, I just have to upload it in the YouTube. You can just type my name and then PMRF. Even you just, I think one, it will come up, I guess. So you just have to type PMRF thing, PMRF, and then so when this it comes up, it will show. So that's the thing. I think I am going a bit slow because I'm covering this concepts. I don't know if it will be useful for you right now, but I know that this concepts will be extremely useful during your interviews, and then this competitive exams like CSI and DBT, they mostly ask this kind of questions. So the question was like, should I continue like this, or like should I just stick to this questioning answer session? I had prepared a lot of questions here, so I have a lot of questions to solve. And around 60 questions. So like, should I prefer like which thing? Like, should I just teach things, or like the question answer session is good enough for you guys? So I have like some kind of questions prepared in this as well. Okay. Maybe both of them. I'll try to fasten up my teaching thing, like so that from next time, like, but it's good if I let you guys think a bit before answering, or like so that you guys can remember things properly. Mm, okay. I'll try like maybe next I have to go now, but maybe from the next class I'll try to speed up things. Like I will not give take lecture method. So anytime. Okay. So lecture method so that any type of question can be answered as yeah, like yeah, I think like this way it's quite good, but I think I'm a bit slow while teaching. So maybe next time, okay, like I'll try to teach in next class, maybe next Sunday, I'll try to complete all this glycolysis and then we'll start with what you guys are learning right now. What is week 8? Actually, I've not seen that. What is week 8? It's Krebs cycle or something else. Okay, maybe I'll check it. Like, if I look, I'll check it. Uh, I'll try to finish this thing like glycolysis as well as Krebs cycle in next term itself. Oxidation. Okay. Next class, I'll try to finish this faster within 1 hour, and then next 30 minutes, like not 1 hour, 1 hour is quite less, 1.5 hours, and then later in last 30 session, like 30 minutes, I'll ask you some questions and I'll try to solve some MCQs. In that way, you guys can get hold of it. I think still have 5 minutes. If you guys want to answer, like we can do some questions. So I have some questions framed on like this. Okay, let's answer like some questions. Can you please share PDF? Can you please share PDF of questions? You guys don't have that link. Okay, maybe okay. Like in the, I can provide you the link of this folder itself. Actually, we have this link, but I think it is only shared with the PMRF people. Like I mean, the main authority of PMRF. But I'll send the link in that thing. The YouTube channel that I have. I'll just show you guys. So you can just type some. So actually, it's mandatory for us to record these sessions and then upload it. Um, to upload it to the YouTube in YouTube as a proof that we are involved in teaching. So yeah, this is the channel. Like yeah, yesterday whatever I taught it was uploaded here. I have to upload, so you guys can see check it out. So like whatever I have taught till now. So till there is a till sixth session. Today there is a seventh one. So I'll upload it here and then I'll um, mention the link of the PDF thing. Your screen is not visible. Is it visible now?
Yes.
Yeah, so like basically it's in the YouTube. Like you can just go here. So as I said that we are like it's mandatory for us to upload all these recorded videos. So basically that's why I need to record all these sessions and then I basically upload it here. So you can find all the sessions. If you guys have any doubt, you can ask in the comment section if you want, or like in the next class by Sunday that we will meet at the same time. I can clear that. So you just have to type PMRF, my name, and in this text box, the link I'll share the that link. Which link? In every video, under every video I'll just paste the Google folder link. So so this was yesterday's session. So basically I'll just put it here in the introduction of this channel. I'll just put the PDF link, sorry, I mean the Google folder link. and then you guys can download all the PDFs from there. Um, so like just for 10-15 minutes, like 10-15 minutes, let's solve something. Like let's see if you guys have understood something. I'm not that good in teaching right now. It's my first time that I'm teaching here. Let's see. So let's go with this 45th question. What is the first step in glycolysis and what is the enzyme responsible for this step? So I think this is one of the easiest question, I guess. So you guys can answer directly.
A.
So the answer, like, this is one of the easiest question. It's the answer is A. So second thing is what is the end product of the energy releasing phase of glycolysis? What is the end product of glycolysis? Just A. So when glycolysis gets completed,
D.
yeah, it's D. So when glycolysis is completed, we get pyruvate, we get ATP, and we get NADH as well. So the answer is all the above. So next one is which enzyme catalyzes the conversion of, okay, this I haven't thought, but if you like, this is just a normal thing, I guess. Like there is this memorizing part. So I think you guys, I'm trying to like make this glycolysis part a bit interesting by telling a lot of facts, because initially it was all about memorizing thing. Okay, what is this? Okay, answer the question number 50. Which step in glycolysis is regulated by ATP and citrate cycle, citrate levels?
B.
It's the fructose 6 phosphate to fructose 1,6. Okay. The answer is B. So B was the answer. Okay. Sorry, like it was, I didn't edit this. Okay. Next one is which enzyme catalyzes the conversion of, okay, these things from whatever I have taught. It's not there. So we all know that answer is hexokinase here. I need to prepare some interesting questions. Directly, which of the following molecules can bypass the first step of glycolysis and interact with the glycolytic level? Up to 6 months. Can anyone answer this one? 64th. Which of the following molecule can bypass the first step of glycolysis and enter directly into glycolytic pathway at the
D.
level of fructose 6 phosphate? It's 6. Mannose 6 phosphate. Now there is this pathway, like, which was that pathway? I forgot. Like one pathway is there. Okay, like maybe I'll frame some other questions here. Like these are normal questions that we study. Okay, like I have to end this question like session now. Sorry, like next time, like mostly we will decide. What we will do is, as you guys suggested, we will go with this thing. So next time, what we will do is we'll go with uh, all the questions as well as the which thing questions, as well as I will continue to teach you all these concepts, and next time I'll be like a bit quick so that we can cover all this session, like I mean all the concepts in one go. So next time I'll just bring some questions as well as this thing as well. Okay, then like we can, you guys can log out. We'll discuss in the next session. Thank you.